welcome back. This is Sans Lectures. If you've not liked, subscribe and followed, please do so. We are still continuing with our cell communication series. Last time we discussed the essence of cell signaling, cell communication. We began on cell signaling, we saw the various types of cell signaling, and today we are going to look at receptors. If you've not yet seen those, please go look out for the video. So today we are going to look at receptors. There are two types of receptors. We have what they call intracellular receptors, and we have what they call extracellular receptors, or what they call membrane bound receptors. So let's first look at intracellular receptors. Intra means inside. So they are located inside the cell. They are found inside the cell. Inside the cell, that means they are suspended within the cytoplasm. That is why when you read some text, they will be calling them cytoplasmic receptors. Another important thing to note about them, they bind hydrophobic molecules. So what does being hydrophobic mean? Water hating. So they bind water hating molecules. That means these water hating molecules can easily pass through the cell membrane. From our first videos about the ultrastructure of the cell, we saw the cell membrane, and we say the cell membrane is made up of a phospholipid bilayer. That phospholipid bilayer prevents all hydrophilic molecules from passing through. However, hydrophobic molecules can pass through, and those hydrophobic molecules are usually small in size. So they can first via the hydrophilic heads, and they pass through the hydrophobic tails until when they enter the cytoplasm. Then within the cytoplasm, they look for their receptors. Examples of intracellular receptors will include testosterone receptors. We know that testosterone is a molecule that is made out of cholesterol. So it can easily cross the cell membrane. Then we have estrogen receptors, and all other molecules that can easily cross the cell membrane have their receptors within the cell. Let's continue with extracellular receptors, or what we called membrane bound receptors. These ones they have an extrinsic site, they have an intrinsic site, and they have the one that spans across the membrane. So if this is our cell molecule, our cell membrane, they shall have a site that spans across the membrane. They shall have a site that is found within the cell membrane, and they shall have a receptor that is found outside the membrane. Membrane-bound receptors majorly bind hydrophilic molecules. They bind hydrophilic molecules, which molecules cannot easily cross the cell membrane. So they bind to receptors on the cell surface membrane and will trigger a cellular response, as we are going to be seeing. I say they bind hydrophilic molecules, and there are three types of membrane bound receptors. Number one, you have the coupled protein receptors. Number 2, we have the enzyme coupled. We have the enzyme coupled receptors. Enzyme coupled receptors. And number 3, we have the ion-gated channels. Ion-gated channels. So we have the G-coupled protein receptors, the enzyme-coupled receptors, and the ion-gated channels, or what, when you read in subtext, they will call the ion channels. The ion channels. Yes, after us having had a very brief background of membrane-bound receptors, let us look at the first type of membrane-bound receptors, which are the G-coupled protein receptors. So they are G-coupled protein receptors. I said a characteristic of membrane-bound receptor, it has an extrinsic site, the one that spans across the membrane, and an intrinsic site. So our G-coupled protein receptor, we have a receptor, that is why we call it a G-coupled protein receptor. It will have a receptor on the cell surface membrane. Then we shall have our G protein, which is our intrinsic site in this case, and we shall have a membrane that spans 7 times. We shall have a protein that spans 7 times across the membrane. Across the membrane. When I say 7 times, I mean 1, 2, 3, 4, 5, 6, 7. So it spans 7 times across the membrane. And G-coupled protein receptors are called so because they are bound to the G protein, which G protein is called the guanonucleoside binding protein. The guanonucleoside binding protein, which is our G protein here. And this G protein is made up of 3 sub-units. Number 1 is the alpha unit. The alpha unit, why did I label them here? This is our alpha unit. This is our beta unit. And this is the gamma unit. The gamma unit. And as we said, it is a G protein. It is always bound with GDP. In case there is no signaling molecule that is intercepted on the cell surface, it is always bound with GDP. GDP is guanoside dinucleotide phosphate. So we say typical of any signaling pathway, we shall have reception, transduction, and cellular response. So I'm assuming my signaling molecule comes and binds on our receptor on the cell surface membrane. It shall trigger a transduction pathway. For G-coupled protein receptors, our transduction pathway shall involve activation of this cytosolic tail. This is what you call the cytosolic tail of the intrinsic protein, the cytosolic tail of the protein. Yes, then it shall also have our GDP here being replaced by GTP that is fully floating in the cytoplasm. So this GTP shall come here and replace the GDP. However, one thing I need you to note is that GDP or GTP always binds on the alpha unit. That is why it is the alpha unit that will interact with other molecules. Another thing important to note, we have 3 types of G-coupled protein receptors. We have GS, we have GI, and GQ. But for purposes of learning today, we are going to look at the GQ and GS. Yes, the GQ is, that means it is guanonucleoside coupled protein receptors, but specifically of Q. Since we shall have, they function in such a that we shall have a receptor binding on the cell surface membrane, the cytosolic tail shall be activated, then the alpha unit shall have GDP being replaced by GTP, then the alpha unit there shall interact with another very important molecule called phospholipase C. To interact with phospholipase C, a molecule that is usually located next to it. So we shall have our phospholipase C molecule here. Then phospholipase C shall cleave another molecule, another enzyme called PIP2, phosphatidyl inositol biphosphate, which is usually here. So this PIP2 shall be cleaved into two important molecules. Well, number one is DAG, which is diacyl glycerol. Then number two, we shall form IP3, which is inositol triphosphate. This was phosphatidyl inositol biphosphate. This is inositol triphosphate. Then inositol triphosphate is a small molecule. It's a small molecule that is soluble across the cytoplasm. While this diacyl glycerol, or what is summarized as DAG, remains attached to the cell membrane. So what happens is this. This IP3 shall move across the cytoplasm until when it reaches the endoplasmic reticulum, specifically the smooth endoplasmic reticulum. We are told that the smooth endoplasmic reticulum is the store of calcium ions. So the smooth endoplasmic reticulum has receptors that can bind IP3. And when IP3 is bound, it will cause the release of calcium ions. Which is our first cellular response in this case. So when calcium ions are released, they can trigger different pathways, number like depolarization, which depolarization is very, very important in generation of impulses. However, these calcium ions can also activate this other part, diacyl glycerol, which diacyl glycerol will then bind with protein kinase C, which protein kinase C can activate other proteins by phosphorylation. Phosphorylation simply meaning addition of a phosphate. I repeat, when a signaling molecule binds on the receptor, the cytosolic tail becomes activated. Then also GDP is replaced by GTP. This causes the alpha unit to interact with phospholipase C. Phospholipase C will then cleave PIP2 into IP3, which is inositol triphosphate, and diacyl glycerol, which is DAG. IP3 is very soluble, so it will move through the cytoplasm until when it binds to its receptors on the endoplasmic reticulum. We say it's strictly a smooth endoplasmic reticulum. Why? Because it is a store of calcium ions. Whenever it binds, there is release of calcium ions. These calcium ions cause various cellular response. Number one, depolarization, which depolarization can lead to generation of impulses. However, the calcium ions can also activate DAG, which DAG interacts with protein kinase C, which kinase C can phosphorylate other proteins, triggering further cellular response.